Thank you very much, Francois, and I'd like to thank Rob for the kind invitation to come and talk at this meeting. I have to say I've really enjoyed listening to the talks so far. I've learned a lot, so thank you very much, and it helps me to kind of position what we're trying to do in UCD, and maybe what Peter said earlier on, I hope is reflected a little bit in what I'm going to talk about. So to just kind of kick off with some background, and I suppose my approach here is a little bit more academic than what you've heard so far, but nevertheless I'm going to try to kind of keep the concepts that Peter mentioned, which is this fundamental idea and this applied idea and the solution type approach in mind. So as Francois said, we've been working on kind of low moisture and the microbiology of low moisture for quite some time. And I suppose, you know, many of you are far better educated in the audience than I am about low moisture foods. So some of these earlier slides are kind of like teaching my granny to suck eggs again. So I don't want to insult my audience. So apologies if I attempt such. But again, we've seen the use of drying methods, you know, as a traditional approach to preserving food. And we know, for example, that dried foods, the idea here is to try and lengthen the shelf life by removing moisture, and that has obviously a direct impact on what we call the water activity. And typically then, you know, as Marianne was alluding to somewhat this morning in her talk, although she didn't explicitly state it, there are different kinds of low moisture foods, many of which include things like nuts, cereals, and honeys. And I suppose, you know, when I talk to students in class and sort of ask them, you know, what do you think is your perceived risk associated with the low moisture food, they might say, well, actually, it's probably minimal. And I suppose if you went out onto the street here in Amsterdam and asked 10 people, they'd probably say the same thing. But we now know that that's actually not true. And we know that despite the fact that these types of food matrices cannot support the growth of bacteria, bacteria can survive or they can persist. And therein lies the problem, is to try to understand the nature of that persistence. So perhaps that requires some fundamental investigation and then a translation of that fundamental outcome into an applied kind of an approach, if I take Peter's point earlier this morning, and then the use of that applied approach for risk management and the subsequent, hopefully, alleviation of the problem. So in terms of talking about the kinds of research I'm going to talk about, I always keep in mind that there's got to be some application down the line which helps a risk manager to translate into effective controls. You know all the ways in which we can remove moisture from foods, and that's fine. However, what we don't sometimes think about is the consequential effect of that when we consider the microbial population. So here are some of the consequences that at least I can think of when we start to remove moisture from the environment in which a bacterium may find itself. For example, the metabolic activity of an organism changes, and we can see this, and I'll show it to you a little bit later. We know, for example, that spores can tolerate these types of environments, and actually vegetative cells can also adapt and remain viable. And again, the two typical examples from the point of view of the dairy industry would be Salmonella and Chronobacter as being very good proponents of this. And then, of course, there's also this risk of cross-contamination of low moisture foods that can arise from exposures in what we call the built production environment. And again, it's very interesting to hear the views expressed this morning in terms of taking a total look at your production site, not just, if you like, the processing chain itself, but the environment surrounding that processing chain, which can potentially influence the final product. So to that extent then, we need to think about this slightly differently, and we need to kind of put ourselves in the mind of a bacterium in terms of what it is trying to do here in order to alleviate and overcome the stresses that we as the controllers are going to impose. So we need to start understanding a little more about the bacterial adaptation then under these conditions and how do they do it. And for this, I'm going to show you the use of whole genome sequencing, or a type of whole genome sequencing that will help us to start at least to tease apart some of these issues. So we know, for example, that low moisture foods are not sterile, but yet we've seen now increasing numbers of outbreaks being reported. And again, as Francois said earlier on, there are plenty of reviews out there that summarize this kind of epidemiological information. We still know very little about how bacteria are behaving under these conditions. And if we're going to try to control them, surely to God, it means we have to start understanding how they're behaving. the hygienic protocols and the manufacturing processes under low moisture conditions may not necessarily be fit for purpose, although I'm open to correction on this, in terms of trying to deal specifically with this issue or this challenge. And then, as I've said a moment ago, the main pathogens of concern, of course, are Chronobacter species, and we've had a really good example of that over the last number of months with the Sturgis plant in the US and the things that can go wrong. And of course, more locally here, the Ferrero issue with respect to Salmonella. And so these two bacteria in particular present this unique challenge insofar as we're currently situated. So let me just take an aside for a second. I just want to do a side swipe for 1 second because I want to show you something. So this is a picture of the Entero database, just looking at all Salmonella serovars. There's about 32,000 entries here, pre-2011. This is from a paper by Mark Ackman, which I was involved with, and many, many other people over the last while in producing. And I've specifically highlighted the Mandaka clade at about 9 o'clock on this map, so with the yellow arrow. So if we take a slightly closer look at this Mandaka clade, this is what it looks like. So we just take out all those strains and sort of genetically compare them. This is the kind of radial phylogenetic tree that you see here. And if you look at the dark blue, kind of from about 10 o'clock to 7 o'clock, and you look at the light blue from about 11 o'clock to 3 o'clock or 2 o'clock, these are collections of isolates that are Irish. So they're not necessarily related to each other. And so if you delve a little bit further, one of those branches has done this. So as I said to a few people over lunch, this is a story about a Salmonella ambandaka that is colonizing a production site, total colonization of a production site. The bacterium has adapted to that production site. Now, if I ask most of you in the audience the question as to what the lac phenotype for a Salmonella strain is, you'll tell me, I hope, that it's negative. But that's not the case here. So if you look here, this adaptation has caused a movement of genes from Leclercia, which is a bacterium on the top of my slide, and you can see this ICE element, which is an integrative conjugative element, which is a massive chunk of DNA, which carries genes associated with plasmids for transfer, but also genes associated with phages. So it kind of behaves as a hybrid. But you'll also notice in this particular ICE element the typical lac ZYA nomenclature of the lac operon. So what's happened here is this Salmonella ambandaka strain has colonized this particular production site, and it has acquired a complete, intact, fully functional lactose operon that allows it to survive in a dairy processing environment, making it very difficult to get rid of it. And you can see this very nicely by way of the insertion here. So whole genome sequencing can tell you a little bit about the adaptation of some of these strains if you care to look. And so going back to script again, this brings into mind then, this idea of precision food safety. So this was a term that was coined by Marty Weidman and his group in Cornell. And I rather like this because it really does capture for us what we're trying to do here in terms of gaining deeper understanding of the nature of bacteria, whether they're pathogens or probiotic strains, and then trying to use that for whatever the intended purpose is. But here you can see that when we talk about precision food safety, we're mainly talking about the use of whole genome sequencing and the interpretation of that data. And the things we can do with it, for example, are such that we can make accurate identifications of organisms. I think this is hugely important. And we've seen lots of examples of this historically where bacteria are misidentified, and that causes major problems for both companies like yourselves, but also for the regulator. The other thing you can do with this, of course, is you can decode that genome information, just like I showed you with the Mbandaka example, and you can see what the genetic makeup of the strain is in terms of markers or genes that might be relevant to the protection of public health, such as antimicrobial resistance markers or virulence markers. Or if you're not interested in pathogens, you could look at other types of markers that might, for example, tell you something about how the bacteria might behave under a growing condition, such as the ones that Marianne was talking about just before lunch. Epidemiology, I think, is relatively straightforward. Tracking and tracing, hugely important. And I think the holy grail here, and nobody has really mentioned it yet, but the holy grail here, I think, is risk assessment at the level of the genome. So this is where we're using genetic sequences of unique bacteria to distinguish between what I call pathogens that will cause true infections in susceptible humans versus other forms of those bacteria which will not mount the same type of infection. And we've already seen some evidence of this in respect of Listeria monocytogenes, particularly where the internalin A and internalin B genes may be disrupted, and I just don't have time to talk about that. So this is kind of the precision food safety we're talking about, and we have the technology right today to address this. Now, without the risk of sort of boring you to death, I should now start to talk about whole genome sequencing, but we've already heard a little bit about this from some of the other speakers, so I'm not really going to say very much. There's a couple of revolutions of whole genome sequencing here in terms of the various technologies. Most of where we're situated at the moment is with using these benchtop sequencers such as the Illumina MiSeq. And then, of course, you've probably heard about PacBio, which is single molecule sequencing, and similarly Oxford Nanopore Technologies, which is also single molecule sequencing. And what those methodologies do is they sequence DNA, of course, but they do it slightly differently and with differences in terms of the accuracy of the reads that you get. So typically with an Illumina MiSeq, you get up to about 300 base pairs by way of reads. And as you can see, with Pacific Biosciences or PacBio and Oxford Nanopore, you considerably lengthen the reads that you get. So the potential here is that you can use those second two strategies as a means of closing a genome if you so wished. In our lab in Dublin, we use both the MiSeq and a MinINE. So we use MiSeq because it's highly accurate, but the reads are short, whereas the MinINE is compromised in terms of accuracy. The read lengths are long, and so we can use hybrid assemblies of both to reconstruct the genome to give us what we call a closed genome. Now, why is this important? Well, it's important for several reasons, and of course one of them is obviously Genome Tracker. And I'm sure as many of you in the audience know, you're very well aware of what Genome Tracker is and what it's purporting to do. And I suppose we have this debate currently as to whether we should submit sequences to Genome Tracker or hold off. So many of the bigger food companies are probably a little bit cagey in terms of how they want to react to this, but they're certainly doing their own internal in-house sequencing using their own pipelines. And perhaps the beauty of Genome Tracker is you can actually dip your toe into Genome Tracker without ever sharing information with them. So you can kind of get a look and see what's out there with your sequences without ever having to upload your sequences. But in broad terms, I think the principle of Genome Tracker is a good one in terms of trying to prevent outbreak and detect outbreaks before they actually happen. And there's lots of labs all around the world who play a part in this. This has become so active in the last number of years that we've seen an exponential rise in the number of entries to Genome Tracker. And it started out with Salmonella, which is indicated here by the red bar, and you also see here other representatives of sequences that are included here, and included here as well are Chronobacter sequences, as you might guess. Using raw sequence data, as I said, we can interrogate it, as I showed you in the precision food safety slide earlier on, but this is just a more detailed view as to how you would do this using different kinds of pipelines to get that kind of information. And again, you could sort of consider some of this as being relevant or otherwise. So to turn our attention then to the remainder of the talk and the subject matter of the talk, I was asked to talk about Chronobacter species. And so just by way of a reminder, more so than anything else, it's an organism, or a bacterium I should say, which was originally included in the genus Enterobacter, which is a really bad place to find yourself because the Enterobacter is a dumping ground for bacteria when we really don't know what they are. But some years ago, in the early parts of 2000, a postdoc in my lab, Dr. Carl Iverson, in collaboration with colleagues in Nestle and Professor Roger Stephan at Zurich, we decided to take a look at the genus of this particular bacterial species. And when we looked at 16S ribosomal RNA sequencing, the full-length sequencing, we could clearly see that not all so-called Enterobacter sakazaki strains were the same, and in fact they clustered slightly differently. And that's what gave us the idea to suggest or to propose a new genus. The new genus was called Chronobacter, and if you don't know the story is that the origin of that name comes from Greek mythology, from the god Cronus, and Cronus had a penchant for eating young children when they were born so that they wouldn't usurp his power. So a bit of a stretch here, but anyway, it got accepted. And of course, when it got accepted, we then had the chance to rename all of the species, and at the time when we did this work, we came up with 6 species names. There's now 7 species, and one of those, I'm thankful to say, happened to be Chronobacter dubliniensis, which is probably on the lesser scale of evil here. But anyway, when you look at the phenotypes associated The one that really stands out a million miles, to my mind anyway, is its desiccation tolerance, or its zero tolerance, if we want to call it technically. So this is its ability to withstand dry environments for very long periods of time. Now, we don't see a whole lot of infection here, but of course, needless to say, when you do get stories of it happening, it's usually devastating, as is the case in Abbott and other examples before then. And the typical sort of clinical presentations you see are things like leaky gut environment, like necrotizing enterocolitis, and then the bug crosses the gut barrier to cause infections in the blood, so you get a bacteremia and a sepsis possibly. And then if certain strains present at the blood-brain barrier, they may cross and cause a meningitis, in which case 80% of those infections are fatal. So we also see, rather interestingly, infections in older people, people like me, for example, who might have an issue with our swallowing mechanism. And we would also take medicated foods, and the same type of risk is presented. But we tend to think about this just in the context of neonates, and I guess that's very emotive, I suppose, when you look at it. But you know, old people also can get infected with this organism if they happen to be using medicated foods. Same types of risk features. However, in young children, this infection is devastating. In older people, you're likely to end up with things like wound infections or urinary tract infections. So coming back then again to the adaptation that I mentioned, and when we think about this in the context of different environmental niches, and that could be anything. So if we look at the factory, for example, or the built environment, so-called, we might be interested to sort of think about things like biofilm formation. So we know that this is a potential feature that bacteria can express in a production environment that may go some way to sustaining their colonization of that particular niche, if you like. Then there's the adaptation to high temperatures. I mean, some years ago, we measured some of the thermal tolerance of these strains, and they are more thermal tolerant compared to other Enterobacteriaceae. There's also tolerance to acidic conditions, and then there's osmotic tolerance. And osmotic tolerance, I think, becomes key here. If you then look at this and you swing around to the left-hand side of my schematic here to the infection strategy, then we think about other types of adaptation, like motility, for example. Quorum sensing might be an issue here. Things like the ability to adhere and to colonize and translocate across that gut barrier. And then when you get deeper into the human host, the susceptible human host, there's sort of considerations with respect to systemic persistence, such as the evasion of the immune system, or indeed macrophage survival. So suffice it to say, the reason we called this organism, way back when we mentioned it, the reason we call it Chronobacter species was to try to encapsulate all of these features so that we wouldn't confuse risk managers in terms of trying to see, well, is it a Sakazaki or is it a Melanoticus, and is the profile different? We didn't want to confuse people by looking at that. So we assumed that every single Chronobacter has equal potential to mount an infection in a susceptible human. And of course, the way that happens, and it's not like a baby like this, but the baby usually consumes contaminated feed, where the numbers of bacteria we assume are very high. The organism transits through the alimentary canal to the gastrointestinal barrier, where, as I said, if it crosses, you get this necrotizing enterocolitis, on into the vasculature system, and then deeper systemically until it presents at the blood-brain barrier. And we know already there's one particular biomarker, which is this outer membrane protein, or this porin A, as it's called, which seems to be important in locking on to the receptors of the meninges and allowing the organism to cross. Coming back again to the adaptation in the factory environment and the contamination routes. Well, Jeff Farber produced this figure a long time ago, but I think it's very useful to kind of remind us in terms of the kinds of challenges we're facing both in the production environment, but also to some extent the challenges we're facing in the home or the care environment. So if we look at this in the context of intrinsic contamination, which is when we get contamination in the production environment, there's a couple of challenges that Chronobacter needs to mount. One, of course, is dry heat. But I've already said this is reasonably thermal tolerant, so that seems pretty okay. Low moisture is another one, which is obviously the manner in which many of these production sites are run. You have limited nutrients, you would use sanitizers in the environment, and then you have to think about the microbiome of the environment, and does that have any impact on the colonization potential for Chronobacter? And we don't know the answer to that just yet. So when you transit then the production site, you're out into the home or hospital environment where extrinsic contamination can happen, and this could be as a result of poor hygienic measures in those particular locations that cause contamination and subsequently then cause infections. But as I say, this is a ubiquitous organism. We don't really know what the origin of it is, but we think it's associated with things like plants or dried herbs. And this is a recent paper from Ben Tall's group at FDA. And what Ben showed here is that, in this particular collection of isolates, these all came from dried plants or plant material associated. And you can see that there's a wide range of heterogeneity, if you like, by way of this radial tree here. On the right-hand side of my slide, then, you have an infection model to look at some of the representative isolates from the tree in terms of their behaviors in these zebra embryo fish models. And you can see here from the green bars that only 2 of those green bars touch 100%. So one of them is this 29544 strain, which is the extreme left of that bar chart, and that's a sequence type 8 strain. The other one is a sequence type 17 strain, which also touches the 100% bar. If you look at the sequence type 4 bar towards the middle of that bar chart, that's interesting. Sequence type 4 clinically is supposed to be the strain that causes the majority of human infections. But that's not exactly how you would decode that in terms of what this infection model is telling you. And then, of course, we saw during the past few months the inevitable downsides of some of these consequences. And the report from CDC of this Chronobacter contamination of powdered infant formula coming from the Sturgis production site in Michigan. And at the time when I made this slide, there were 4 recorded illnesses, 2 deaths. And, it begs the question, why do these events happen and how do they happen? Well, if we go back again to the physiology of the bacterium, there are some things we do know. So, for example, Chronobacter is no different from E. coli or Salmonella in terms of its behavior at low water activity. So we need a water activity here of about 0.94 for this bug to grow. We've already said it's relatively resistant to heat, but when you dry this organism down, all bets are off. The physiological behavior changes quite dramatically. It increases tolerance to heat, it increases tolerance to biocides. So the very kinds of measures we're using to try to eradicate it, we have to sort of be cognizant of. And then, of course, because of this desiccation tolerance that it exhibits, it has this propensity to survive for weeks, months, and even years in low moisture foods. Now, some time ago, when we used to work with what was then called Royal Dutch Pneumocco here in the Netherlands, we produced this slide to their board, which then drew several gasps from people. So this was a surveillance using pulsed field gel electrophoresis at the time of one production site over a long period of time. And you don't have to be a genius to figure out what's happening here, because what you can see here from these pulsotypes is that we have one strain or one pulsotype that appears to be common. It doesn't disappear, it appears to be persisting within this production environment. So understanding, as Peter was saying earlier on, understanding the dynamics of this environment is hugely important in terms of your ability to manipulate and control it. And this was a particular strain that interested us. And the question was, why does it persist? The subsequent upshot of a lot of this work gave rise to a PulseNet subtyping protocol at the time, which was on the PulseNet books. Of course, this is now all whole genome sequencing, but this was still relevant at the time when this work was done. And that SP291 isolate, which I indicated towards the top of the dendrogram with the blue arrow, was the strain in question. So we took a look at this over a very long period of time, and we compared its ability to survive desiccation against other strains that we would expect to find in the same kind of environmental niche. And if you look at the graph here, and particularly the turquoise line towards the top, that's the survival curve of Chronobacter sakazaki SP291 for a period of 32 months. So you get just over a 2-log reduction in powdered infant formula. So this strain is undoubtedly adapted to powdered infant formula and surviving in the production site. The question was, how does it do it? To address this, we started to look at this by way of what we call an RNA-seq experiment. So this is an experiment which, instead of sequencing the DNA, we're now sequencing the RNA. So we're sequencing the genes that are expressed when we select this bacterium under a particular environmental condition. So before I explain it to you, if you just look to the top right-hand side of the slide here, you get the typical blue line, which is the growth curve of the bacterium over time. And I've indicated with the pink arrow here the early stationary phase. So we reckoned that that would probably be what the organism might look like in a factory environment at a time. And then the inset curves are desiccation curves. And the red line here is the desiccation curve of sp291 in powdered infant formula. And you can see that at least over a period of about 21 hours, you hardly get a reduction in the log number. So again, it sort of confirmed our hunch from the earlier slide that this strain seems to be adapted to this environment. If you desiccate sp291 in TSB, for example, the green line, or in water, the purple line, you see that you do get a reduction in cell number. So I suppose the nature of the matrix itself supports the survival of this strain. So the experiment here is as follows. We took Chronobacter sp291, grew it to early stationary phase, split the culture, and then took RNA from the early stationary phase, and then we desiccated the other half of the culture onto stainless steel coupons, exactly the same type of coupons that were in the fabric of this particular production plant. We recovered the total RNA, we sequenced it, and then we mapped every single read back against the original sp291 genome, and you get a representation of a statistical distribution here from gene to gene and in the intergenic regions. And then we start to interpret that using statistical programs to identify the numbers of genes that are upregulated. And you can see here from the pie chart that there are about 115 highly upregulated, highly downregulated and highly upregulated about 365. Most of the genes don't do anything. So they just don't change, neither up, neither down. Looking at this slightly differently, we can see it as follows. So if you look at this slide, the midsection of the slide is the actual DNA strand, and you're looking at the piece on the top as the sense strand, so-called, the piece on the bottom as the antisense strand, and you have genes dotted along those strands of DNA, as you know. So what the RNA-seq data shows us is that if you take these two operons, so the operon on the left produces betaine, the operon on the right is a proline transporter. Both of those molecules are what we call osmoprotectants and are used by the bacterial cell to withstand the osmotic shock when you withdraw water. So you can see here in the early stationary phase culture, those genes are switched off. There's no transcriptional activity across those genes. But in the desiccated phase, you can see that the genes are switched on. You get these red peaks, as you can see, because that's the mapping of the reads back against the genome, and you get that sort of distribution. And the red peaks here show you a fourfold upregulation of those genes, so they're really turned on very, very strongly. Other examples of those genes included these two genes here, OTSA and OTSB. And OTSA and OTSB are responsible for manufacturing trehalose within the bacterium. So again, these were two other genes that were highly switched on during this desiccation phase. So what we did is we knocked out those two genes and we just compared the growth curve in the blue bars versus the red bars for the knockouts to show that the knockout didn't alter the physiology of the bacterium. However, if you look to the right-hand side here, you can see again the desiccation curve. When OTSA and OTSB are knocked out, the susceptibility of this strain increases to desiccation. So we think we now at least understand several of the markers that are playing a role in this response. The real question now is, does every Chronobacter do this, and does every Chronobacter respond in the same way? So to try to address that, we went back again, and this time we made a comparison between Sakazaki sp291, which was the environmental isolate, and then a clinical isolate, which is 29544. So exactly the same experiment, done in exactly the same way. And again, without laboring the point, you can see the desiccation curve here on the bottom right. The green line represents the clinical strain 29544, so it doesn't appear to survive desiccation as well as the red line shows you for sp291. But what about the level of gene expression? Well, at least when we look at some selected examples across the genome, there are 4 of them shown to you in these two graphs. You see that Chronobacter sakazaki sp291, which is this environmental origin strain coming from the powdered infant formula production site, expresses these genes at a much higher level compared with the clinical strain. So this, we think, is a type of adaptation that we can see in terms of these strains responding to environmental cues that are then underpinned genetically by genes that are either turned on or turned off in order to allow it to survive in this environment. So I suppose from the perspective then of people in quality control or risk management, These are potential biomarkers we could think about looking at in terms of trying to translate fundamental research into actually applied research in terms of trying to help us to deal with these kinds of strains. Now, all of these slides so far have talked about one specific bug, but what about if we talk about every bug? And so we switch tack a little bit. And now we talk about the microbiome. And I think everybody in the room understands what the microbiome is. And you all also know that if you compare, let's say, the output from culture-independent methods in terms of identifying bacteria versus the traditional culture-based approaches, there's a gulf of a difference between them. So in terms of culture-independent methods, or so-called next-generation sequencing methods, we can take any sample we care to think about, isolate nucleic acid, sequence it to totality, and then run it back against various databases to identify the nature of the sequences that have been read through whatever platform we've used. So this is particularly useful in looking at fermentations and in looking at the dynamic of a fermentation over time. It could be particularly useful to what Marion was talking about this morning in terms of looking at the metagenome of those different plant-associated backgrounds that she was commenting upon. And in fact, it also is very useful in terms of helping us to manipulate this, because we could, for example, put pathogens in or put probiotics in and see what it does to the metagenome by way of the changes in the dynamics of the sequence. We could also use this for biological control, so that instead of being mandated by the regulator to look specifically for Chronobacter or Salmonella, potentially we could use a shotgun sequence approach to look for every bug and to decode that information when Chronobacter is present and compare it to decoded information when Chronobacter is absent, and look for those differences which might be indicative of natural biological control phenomena that might just be happening at the level of the plant. That could be extremely useful because it could immediately remove the need, assuming it's validated, of course, for the use of chemical disinfectants. There's two approaches to how you do this. One is either using a 16S ribosomal RNA sequencing approach, which really is focused just on bacteria. So this would mean recovering the 16S ribosomal RNA gene, either in whole or in part, by way of an amplification, subsequent sequencing, and then blast searching across databases such as Green Genes, Silva, or others to identify the nature of those reads and then to give you the operational taxonomic units to identify the bacteria. So this just, as I said, looks at bacteria. The other way to do it is to use a shotgun metagenomics approach. So you just take the total DNA from the sample and you sequence all of that. And then you run those raw read sequences back through several databases to indicate not just the taxonomy. So this time we get bacteria, we get viruses, we may get fungi, any other biological hazards that are there. But in addition to the taxonomy, we also get functionality. So we get genes associated with various functions like antibiotic resistance or particular metabolic genes that appear to be showing within the shotgun sequences. So this is an approximate workflow of how this is done. But we could think about using this in the context of a powdered infant formula production site. So here are two schematic maps of two production sites in Ireland. One which is an old food plant, the other which is a relatively new food plant. And it's just shown to you here by way of different zones that are indicated in different colors. So we have high care zones, medium care zones, and in some cases the low care zone. So the idea here would be to take swabbed samples to look at the environment and the dynamics of this environment over a long period of time to see if we could use, in this case, 16S ribosomal RNA sequencing to get a picture of what that environment actually looks like. So when we do this, this is what we get. So the top part here shows you the genus, the different genera associated with these different care areas across those two illustrated maps that I showed you in the previous slide. The middle section shows you at the level of the phylum. And you can see here, we can clearly indicate that there are some particular phyla or genera of bacteria that appear to be kind of resident, if that's the right word to use here, within the production site. If you look at the bottom of the slide and the right-hand side, D, this describes what I call the core microbiome of these production sites. So this is the microbiome that does not appear to vary. It's the core. And we would obviously then get influences from external and possibly otherwise that may alter that core microbiome, which we can see by way of sequencing. The Venn diagram on the left-hand side just shows you a summary of that, and I think you can see from the intersection of all of those circles that there's about 24 genera here that appear to be consistently identified. Why is this important? Well, we can now start to translate this in terms of trying to refine our control of that environment by understanding the interactions that are happening. So again, this is just a schematic to try to summarize all of this by way of the high care, low care, and medium care area. And what you're seeing here are controls that the production sites have in place, the movement of people, the movement of equipment. But I suppose if I was to try to summarize this without taking it through it in any greater detail, you would say that in the medium care area, you see a greater influence by way of environmental bacteria on the nature of the microbiome in those parts of the plant, whereas in the high care area, it's slightly different. There's a greater influence by way of the environment associated with human-related bacteria. And again, I think this makes sense. And again, as I say, it may be something that potentially has use from the point of view of a risk management approach. So to conclude then, you can obviously see I'm a big fan of sequencing. And I think I would say things like, we need to use this, it's a great technology, but I think it can help us to support, food safety control measures. So the concept of using it to sort of study our environment seems not unreasonable. That environment and the ecology of that environment, I do think, is particularly important, and understanding that is very relevant to what we're trying to do here. And if we can understand the dynamics of that, then we may be able to spot changes in those dynamic features which indicate that something is about to happen, and therefore we can take control of it before it gets out of hand. This type of an approach will also help us differentiate between what I call persistent and non-persistent or sporadic isolates that may colonize the environment. And again, for those that are persisting, perhaps we could consider approaches like RNA-seq as a fundamental approach to trying to decode what's going on. And I think my prediction would be that precision food safety measures are not that far off when we can use these types of approaches linked to the phenotype and to have a better understanding of how this adaptive response happens within these hostile environments, such that we can provide biomarkers to bioMérieux, and then they can go and make money out of it, and then we will have the ultimate subsequent risk reduction measure. Thank you very much. That's the last slide.